Hello, everybody. It's Mr. Caudill. I hope you're doing well. I'm doing well, I think. One of my favorite days of the year, Martin Luther King Jr.'s birthday celebration in mid-January. Please give a moment to think about all those who have stood up to oppression and subjugation and hatred throughout history. Think about all the books that line your history class wall or all the pages in your history books. These are great people, and I wish we could celebrate all of them. The reason why I say they're great is because they stand up. They stand up for what they believe. They have the guts to not know what's going to happen, and often they're persecuted for it. And we celebrate them because they're different and they're gutsy. And no one cares if you remember anything from high school as far as content, academic content is concerned. It will be in your brain, and you'll layer on it when you go and you study whatever your profession is going to be, or you go to college. But what I want you to leave here with is the ability to critically think, to question everything, to love and to be generous, and to want to better yourself and those around you, and to not stand up for what you know is wrong. So if you feel something is wrong, stand up, say something, bond together with your peers. Don't sit around. And again, I told you this before, you will forget all about me, but I want that to be something that you remember and that you subscribe to. If you haven't emailed to me your chromosome defect presentation, please do so. I would love to see it. I'd love to give you a grade for it. And we'll keep going.
The secrets of the X chromosome. These women are identical twins. They have the same nose, the same hair color, the same eye color. But this one is colorblind for green light, and this one isn't. How is that possible? The answer lies in their genes. For humans, the genetic information that determines our physical traits is stored in 23 pairs of chromosomes in the nucleus of every cell. These chromosomes are made up of proteins and long coiled strands of DNA. Segments of DNA, called genes, tell the cell to build specific proteins, which control its identity and function. For every chromosome pair, one comes from each biological parent. In 22 of these pairs, the chromosomes contain the same set of genes, but may have different versions of those genes. The differences arrive from mutations, which are changes to the genetic sequence that may have occurred many generations ago. Some of those changes have no effect, some cause diseases, and some lead to advantageous adaptations. The result of having two versions of each gene is that you display a combination of your biological parent's traits. But the 23rd pair is unique, and that's the secret behind the one colorblind twin. This pair, called the X and Y chromosomes, influences your biological sex. Most women have two X chromosomes, while most men have one X and one Y. The Y chromosome contains genes for male development and fertility. The X chromosome, on the other hand, contains important genes for things other than sex determination or reproduction, like nervous system development, skeletal muscle function, and the receptors in the eyes that detect green light. Biological males with an XY chromosome pair only get one copy of all these X chromosome genes. So the human body has evolved to function without duplicates. But that creates a problem for people with two X chromosomes. If both X chromosomes produced proteins, as is normal in other chromosomes, development of the embryo would be completely impaired. The solution is X inactivation. This happens early in development when an embryo with two X chromosomes is just a ball of cells. Each cell inactivates one X chromosome. There's a certain degree of randomness to this process. One cell may inactivate the X chromosome from one parent, and another the chromosome from the other parent. The inactive X shrivels into a clump called a bar body and goes silent. Almost none of its genes order proteins to be made. When these early cells divide, each passes on its X inactivation. So some clusters of cells express the maternal X chromosome, while others express the paternal X. If these chromosomes carry different traits, those differences will show up in the cells. This is why calico cats have patches. One X had a gene for orange fur, and the other had a gene for black fur. The pattern of the coat reveals which one stayed active where. Now we can explain our colorblind twin. Both sisters inherited one mutant copy of the green receptor gene and one normally functioning copy. The embryo split into twins before X inactivation, so each twin ended up with a different inactivation pattern. In one, the X chromosome with the normal gene was turned off in the cells that eventually became eyes. Without those genetic instructions, she now can't sense green light and is colorblind. Disorders that are associated with mutations of X chromosome genes, like colorblindness or hemophilia, are often less severe in individuals with two X chromosomes. That's because in someone with one normal and one mutant copy of the gene, only some of their cells would be affected by the mutation. The severity of the disorder depends on which X got turned off and where those cells were. On the other hand, all the cells in someone with only one X chromosome can only express the mutant copy of the gene, if that's what they inherited. There are still unresolved questions about X inactivation, like how some genes on the X chromosome escape inactivation and why inactivation isn't always random. What we do know is that this mechanism is one of the many ways that genes alone don't tell our whole story.
Geckos and grasshoppers, worms and watermelons, elephants and Escherichia coli, man and mushroom. All so different in form and function, but amazingly the same in how their form and function are determined. First, all these organisms are made of one or more cells, and every cell of every living thing on Earth contains all the information it takes to create and duplicate and make variations of itself. That information is stored in a very long but quite simple double molecule called DNA, or deoxyribonucleic acid. And the DNA of every living organism is made of chains of 4 smaller molecules called nucleotides. What dictates the difference between a man and a mushroom is the sequence of these nucleotides in the long DNA chain. The 4 differing nucleotide parts, called bases, are made of a few carbon, oxygen, hydrogen, nitrogen, and phosphorus atoms, and the molecules look like this. And each of these 4 bases is attached to an identical backbone molecule, a sugar called deoxyribose, the D in DNA, and a phosphate group. Let's simplify these nucleotides and show them like this. So a single sequence of nucleotides joined by their common sugars would look like this. And the DNA molecule where such sequences are stored looks like this. But how does a simple molecule dictate the form and function of millions of different living things? You can think of DNA as a great library of information. Information that is used to do one thing and one thing only, direct the building of different protein molecules. And it's the proteins that build the cells and keep them functioning and changing and reproducing. Here's where the familiar word gene comes in. If your DNA is a library of information, a gene is a book in that library. A gene is a segment of the DNA molecule. Let's say your body needs a particular protein, like insulin. To get it, some of your cells send a protein signal through the bloodstream to the cells in your pancreas, where insulin is made. That signal protein tells other proteins in the cell's nuclei to open up a part of the DNA double helix, the insulin gene, and start making insulin proteins. As soon as enough insulin has been produced, another signal protein comes to the pancreas's cells that tells them to stop making insulin. It's like looking up a book in the DNA library about insulin and then putting it back when you're done. There are genes in DNA for visible and invisible things that make up your body, like genes for eye color, protein pigments, for skin color, for hair color, for stopping and starting bone growth, for your blood type, for how many fingers or arms and legs you have, for proteins that influence how long you live. Your DNA probably contains between 25,000 and 40,000 genes, while the DNA of a worm or a plant or a fruit fly contains about 12,000 to 20,000 genes. Some of those genes have quite different sequences of nucleotides than yours, and some are similar to yours. Though it happens infrequently, our own nucleotide sequences can change as the result of spontaneous or environmental damage, which might remove or shift a nucleotide position. This changes the gene involved and can then change the protein. Most of these changes, called mutations, have very little effect on the organism or its descendants. Some are mildly damaging, and a few can make the organism better suited to its environment. It is these tiny changes in DNA gene sequences happening over millions of years that create the differences among living organisms. From geckos to grasshoppers, worms to watermelons, elephants to Escherichia coli, and man to mushroom.
Yay, genetics. I read once, well, first of all, we come from a small founding population of about 50,000 people, and this was about 150,000 years ago. They walked out of Africa, and here we are, 7 billion strong today. I once read that there was one person on the shores of the Mediterranean Sea that had one mutation in the gene that coded for eye color. This was about 10,000 years ago, and that caused a lack of melanin or pigment in the iris of the eye, which caused it to be clearer, let's say blue color or whatever, a very light color. And that was it. Every person today who has blue eyes can be traced back to that one person. I don't know if it was a man or a woman, but you wouldn't think that they would have an advantage there on the shores of the Mediterranean Sea where it's hot, think of Italy or Greece, hot and sunny. And so that individual must have been a part of a group of people that kept going northward into probably Northern Europe, Germany, and then Scandinavia, where nowadays we know having a light eye color up there is actually better because there's so little light in the winter that the lighter color doesn't absorb as much light and more gets into the pupil so that the people can see better. And so there's an advantage up there, but there'd be a disadvantage closer to the equator. I love genetics. We did this a little bit ago. I don't want to nail myself down to a date. The program Introduction to Genetics. Look, I get it. It's a piece of paper from about a decade ago, but I haven't found anything that does it as well. What I mean by it is the Introduction to Genetics. So on the back, there's a hybrid cross problem set. I'd like to go over those problems with you right now. So make sure you have yours out or have a blank piece of paper or just try to understand what you're seeing here. Normally I would have kids do this on the board. Fortunately, you get to listen to me. So I'll try to go through them quickly. Number one says, show all the possible genotypes from the following crosses. Indicate the fractions of each genotype. So when we say these crosses, what those letters are referring to are the different gene combinations for a specific characteristic on a chromosome that mom has or dad has. So in the first one, they look like they're heterozygous. And so we can just pull these down and over. And this might be something that you're really good at from middle school. The second half of number one is big G, little g, or heterozygous, and then homozygous recessive, little g, little g. This is a little g, this is big G, and little g. So when they say the fractions of each of the genotypes, what that means is what proportion of the four is each of the genotypes. We have a 1 to 2 to 1 ratio here. 1 homozygous recessive to 2 heterozygotes to 1 homozygous dominant. And down here in the other Punnett square cross, we have a 2 to 2 ratio, 2 homozygous recessive, 2 heterozygotes. You probably understand. So number two says, if G in the above problem is green apples, that's big G, so that must be the dominant characteristic in the population, and little g stands for red apples. What would be the fractions of each phenotype in the two crosses? So you have to ask yourself, which ones of these are going to be green and red if these represent gene combinations in the offspring? So up here we have a green apple. These will also be green apples because the big G dominates over the little, and then this will be a red apple. So this will be a 3-to-1 phenotypic ratio, green to red apples. Here we have a 1-to-1 ratio of green apples to red apples. Make sure you understand how to do this. Let's go on to number 3. Number 3 says, a brown-eyed mother and a blue-eyed father found that their first child was blue-eyed. What's the genotype of the mother? So brown-eyed mother. So if you recall, we've established that big B we're going to use for the gene that creates the protein that causes brown eyes, little b for blue eyes. This is a little disingenuous, I have to be honest, because what we found now is that there's about 30 different genes that combine for eye color, and that's why you never see someone with blue or brown. You see mixtures of everything when it comes to shades of eye color. But we'll roll with it. This is called Mendelian genetics, which means it's just straight up dominant or recessive. It's not polygenic inheritance, which is when you have a mixture. So brown-eyed mother. So let's imagine that mom's genes are up here. So brown-eyed mother means that mom must have a big B, but we don't know if she's a big B or big B, little b. And dad has blue eyes, and they have a blue-eyed child. So we know the answer now because mom has to donate a little b to this union. Then the answer is mom must be heterozygous for eye color with respect to blue or brown eyes. So her genotype is big B, little b. Number 4 says, what's the chance that the couple, here we go, number 4. What's the chance that the couple in problem 3 would have a second child that had brown eyes? So one thing that you need to understand here is that whenever these genes get together and a Punnett square is created or a union of two people occurs or a baby is born, what has happened previously has no bearing on the combinations of genes which occur at that moment. So if this is mom's genotype, what's the percent chance that they would have a brown-eyed child? Well, let's just go through this. It looks to me like it's 50%, 50-50. All right, we're almost done here. In cats, assume that a spotted coat is dominant to a white coat. So I'm going to use the first letter of the dominant characteristic to be our abbreviation letter here. So this is spotted, and then I'm going to use a lowercase s for white. Don't you wish they used a different letter than like P or B or S? How about, a Q? A Q looks really different, uppercase and lowercase, but these guys don't. A white mother cat gives birth to 2 white kittens and 2 spotted kittens. So mom is white cat, so she has little s. And if there's 2 white kittens that are created, then there must be a little s that's donated by dad, right? But there's also 2 spotted kittens that are created. What are the genotypes and phenotypes of dad? White mom gives birth to 2 white kittens and 2 spotted kittens. Man, I psyched myself out there. Sorry about that. Does this look about right? Dad must be heterozygous, meaning a big S and a little s. Mom must have little s, because she is a white cat. And there's 50-50 offspring here with respect to white or spotted offspring. Yay. Sorry about that. All right, here's the last one. Let's see if I can screw this up. And then we're done. A man with blue eyes marries a brown-eyed woman whose mother had blue eyes. So brown-eyed woman whose mother had blue eyes So she must have a little b from her mom, because her mom, when this woman was created, could only have given her a little b. So she still has that little b, but she expresses brown eyes because the big B is a gene that dominates over little b with respect to creation of pigment to create eye color. A man with blue eyes, so he's got little b, little b. What portion of the children would be expected to have blue eyes? Portion of the children, okay. Is that right? 50%. Show the genotypes of all the people involved. Hope you got that right. Hope this is a good review. You'll certainly see some of these on the exam that we have. Finally, the last thing that we're going to do is we're going to do an assignment here. What I'd like you to do is read for 10 minutes and then answer the questions. I think the answers to the questions are in the back of the book of this book I call the Bird Book. I'd like you to start at page 313 and just read 3 pages. And then there are some questions at the bottom of page 318. I won't try to be fancy and do that. Questions on the bottom of page 318. Just do this on a separate piece of paper. If you'd like to challenge yourself, this is our old AP biology book. I call this, incidentally, this is also a bird, but I call it the Eagle Book. I'd like you to read 2 pages in here, pages 184 to 185, and answer 4 questions on page 187. Your choice. I don't care which one you do. You should see me plagiarize these books by taking pictures of the pages and posting it to Schoology. Thanks for your attention. I hope you're still amazed by genetics, and let's keep going.